Okay, so welcome to this video. In this video what we're going to talk about is the use of homologous recombination, okay, to fix broken replication forks, okay. So homologous recombination for replication fork reactivation is what we're trying to do, okay. So the structure then for this video, what we're going to start off with is we're going to start off by defining what homologous recombination is. Then what we'll do is we'll have a look at what a broken replication fork is. Then we'll see the big picture of how we're going to reactivate a replication fork. And then what we'll do is we'll look at the in detail proteins that are involved in doing the homologous recombination for the replication fork reactivation, and we'll specifically look at those proteins in Escherichia coli, the bacterium. However, the principle is exactly the same throughout all different cells, basically. We've found this in human cells, we've found this in all cells that you can imagine, basically. So this is a very standard technique for fixing broken replication forks. Okay, right. So let's start off by talking about the definition of homologous recombination. Okay, so we'll start off with what is recombination, what is genetic recombination. So basically, genetic recombination means moving large chunks of DNA around within the genome. So let me give you an example. If we have a double-stranded piece of DNA here, okay, and let's have two regions. Let's have this portion here in red, okay, and then let's have this portion here in turquoise. What we could do is we could swap those around. We could move this, we could cut this turquoise bit out here, okay, we could then cut the red portion out and we could swap them around. We could put the turquoise portion in here and put the red portion in there. Okay, so what we'd then end up with is something like this. So let me just draw the two chromosomes again. And now what we've got is our turquoise one is down here. So let's have that here. Okay, and then our red one is up here, like so. Okay, that would be an example of recombination. It's just moving large chunks of DNA around, not necessarily on the same chromosome. It can be within the genome. So remember, in humans we have 23 pairs of chromosomes, so we can move them around within the chromosomes as well, and that would also be considered recombination. Okay, right. So what is homologous recombination? Well, homologous recombination means that the two pieces of DNA that you are exchanging are in some ways similar, basically. They're homologous. Okay, so homologous recombination is more normal within recombination. Okay, it's not common to see huge, great chunks of unrelated DNA being swapped around, basically. Homologous recombination then means that these two pieces of DNA that you are moving around are going to be similar, basically. Okay, so that's all homologous recombination means, that these two pieces of DNA are not going to be too dissimilar. Okay, and in our case, they're going to be pretty much identical, basically. Okay, so let's now turn our attention to what a broken replication fork is. So we'll start now discussing the motivation for why we need some sort of repair. Okay, so what is a broken replication fork? Okay, so let's start by drawing our chromosome here. Okay, so we'll just work all the time with one chromosome here. Now, of course, the two strands of DNA are running anti-parallelly. So let's say that this is the 3' end of this strand, and then this is the 5' end of this strand. And colouring in things here is going to be extremely helpful. So I'm going to colour this strand in blue here. Okay, and then the other strand will be anti-parallel to this one. So it will have its 5' end that side and its 3' end this side. Okay, and we'll colour in this anti-parallel strand in vivid purple here. Okay, so those are our two strands of DNA that are complementary to each other. Okay, let's now say we want to copy this chromosome. Okay, so what will happen is DNA polymerase will start working its way along the DNA from this side, and it will be synthesizing new pieces of DNA that are complementary to both strands. So let's show this. So now what we've done is we've opened the DNA like so. Okay, and this open portion over here is a replication fork. Okay, and we're creating new pieces of DNA which are complementary to these original pieces. Okay, so let's colour these in. So here is our purple strand here. Okay, here is our blue strand here. And now we've created these new pieces which are complementary to both of them. Okay, so we'll colour the one that's complementary to purple in red here. And then we'll colour the one that's complementary to blue in orange here. Okay, right. So this is our replication fork, and it should continue on perfectly happily. However, let's say we're going to have a problem. Okay, and the problem is going to be this. We're going to have a single strand break in one of these pieces of DNA. Okay, so let's say we're going to cut this blue strand here. We're going to break the sugar phosphate backbone. Okay, so we've got a single strand break here, and the less fancy term for a single strand break is that we've got a DNA nick here. People often call the single strand break a nick. Okay, so there's a hole there, effectively. So what's going to happen when the replication fork reaches here? Well, you're going to continue synthesizing the complementary strands of this blue portion, but then, of course, once you've peeled this blue portion away from the pink portion, it's going to completely detach off. So what you're going to end up with is this little blue portion here with a new orange strand that has been completely matched to it. Okay, so this is the blue strand, and here's the orange strand, and then this is going to completely break away from the rest of this. Okay, then what you'll have is the rest of the blue strand, which will continue on here, which will now be completely detached. Okay, so here's the rest of that blue strand. Then we've got the purple strand here. Okay, like so. And now we've synthesized that red strand up to the point where we've got this blue strand here. So we've now got our red strand here. Okay, and now that is the concept of a broken replication fork. Okay, we've now got this small little fragment of DNA here. Okay, and then we've got the long rest of the chromosome here. This one's fine. This is a complete copy of this original chromosome here, but now we've got this tiny little piece here. So we're imagining that we've just started the replication here. So we're just at the start of our chromosome here, synthesizing the new strands, and then we've hit this DNA nick here, and this has caused the replication fork to break. Okay, so this is a broken replication fork that we've got here. Okay, so how are we going to fix this? That's the question. Okay, well, we're going to use homologous recombination. Now, this gets quite complicated. Okay, so I'm going to start by giving you the big picture of basically what we're going to do. Okay, so the basic answer to how to fix this is this orange strand here is complementary to this red strand here. Okay, this blue strand here is complementary to that purple strand there. So why don't we take this strand here, reopen it up? Okay, so let's reopen it up like so. Okay, so I'll draw this. So here is the purple strand here. Okay, so this is purple here. And we've now pulled the purple portion apart from the red portion. Okay, so this is the red portion here. And then we've got the blue portion here of that second full strand here. And now what we're doing is we're going to match this orange strand to the red strand here. So we're going to put that in there. Okay, like so. And then we'll match the blue strand to the purple strand. Like so. And then we can continue on the replication. Now you'll notice this is not a full repair. Well, we haven't gone, well, it will be fine, it will work, but we haven't gone back to what we originally had. We have moved the strands around, basically. For instance, this blue strand that's now forming part of this new complementary strand, this was originally part of one of the original strands, and that red portion, that was originally one of the ones that we were copying. So we have swapped things around here, but that's the basic answer of what we're going to do. Okay, we're going to reactivate the replication fork like this. It's a little bit more complicated than that, as we'll come to, but that's the big picture of how we're going to fix this problem, basically. Okay, right, so now we're going to work our way down. Okay, so that was the biggest picture of them all. We're going to go to a new picture which is more accurate, which still isn't the most in detail we're going to go. Okay, so we're going to have 3 levels of pictures. We're going to start with this massive picture here, which just shows you the basic idea. Then we're going to work our way down to a slightly more in detail picture, and then we're going to go even further. After that, we'll go to an even more in-depth picture. Okay, so in this next picture, what I'm going to do is I'm just going to show you what we're going to do, but I'm not going to show you which proteins are going to do it. And then in the final picture, what we'll look at is which proteins are actually going to do it. Okay, so let's now draw another picture for this then. So, we'll start off by reminding ourselves of what has happened, because it's always best to do that. So, here is a turquoise strand, and here is a blue strand. So this is our original chromosome that we want to now copy. Okay, once again we'll say that this is the 5' end, this is the 3' end, this is the 5' end of this blue strand, and this is the 3' end of this blue strand. Okay, then we're going to start opening the double-stranded DNA at this side over here. Okay, and we're going to produce a replication fork. So let's draw this. Okay, so here is our turquoise strand here, now opened, and here is our blue strand here, now opened. Okay, so we've produced our replication fork, and we're producing our complementary strand. So we're producing a new strand here which is complementary to the turquoise strand that's in red there, and a new strand here that's complementary to that blue strand. Now let's say that we have some sort of nick here, okay, and that means that we're going to break our replication strand. Okay, so DNA polymerase will work its way along, and then it will gradually peel this turquoise strand away from the blue strand, and then once it's completely peeled it away, of course the turquoise strand will completely detach from this structure here. Okay, so what we'll end up with is that turquoise strand with its newly synthesized complementary strand here in red. Okay, but now it's detached from the rest of the structure, so here's the rest of that turquoise strand here. Okay, it's now got this orange portion making up that gap there, which is complementary to the blue portion, and then the blue portion is the only portion that's been able to remain intact, basically. Okay, right, so that's our broken replication fork. So, what we begin with then is we will see the enzymes that do this in E. coli in a moment, but what we're going to start off with is we're going to start off by trimming the end of this strand here down. Okay, so we're going to start here and we're going to trim down, basically, and we're going to trim both strands down. Okay, so this is the 5' strand. This is the 5' end of the turquoise strand, this is the 3' end of the turquoise strand, this is the 5' end of the red strand, and this is the 3' end. So what originally happens is you trim both of them down, so we're going to cut both of them up and we're going to move backwards, basically, okay, until you reach a certain sequence on the red strand, okay? Until you reach a certain sequence on this strand that's got its 3' end at the side from which we are cutting them back. Okay, so you're going to wait until you find a certain sequence on this red strand here. Okay, and you might ask, well, what's significant about the red strand? Well, it's that this is the strand which has its 3' end at this side where we are working our way along. Okay, so the special sequence that you're going to be looking for is what's known as a chi sequence. Okay, and let me just give you this chi sequence. It's a sequence of 8 organic bases. Okay, and the special sequence is GCT. Okay, and this is in E. coli, by the way. Okay, so this is the principle within E. coli. It will be a different sort of sequence in humans, but the principle is the same. Okay, GCT, GGT, GC. Okay, so, and this is going from the 5' end, so this is the 5' end, and this is the 3' end. Okay, so this little box that I've got here, this is this sequence here. Okay, so to re-amplify this, what's going to happen is an enzyme complex, the name of which we'll see in a moment, is going to latch on here, it's going to work its way along, and it's going to be breaking up both the turquoise strand, which has its 5' end this side, and also the red strand, which has its 3' end this side, until it reaches this chi sequence, which will be somewhere on the red strand. So E. coli has loads of these chi sequences dotted all around its genome. Okay, so it will eventually reach a chi sequence. Then it will stop degrading the red strand, the 3' end strand, and then it will continue degrading the 5' strand for a while. Okay, so what that overall produces is something like so. So we're going to continue degrading the turquoise strand, the 5' end one, even though we've stopped degrading the red one. Okay, so we're going to produce this overhang on the 3' ended strand. So this is the 3' end, this is the 5' end, this is the 5' end of turquoise, this is the 3' end of turquoise. Okay, and this is what's known as a 3' extension, what we've produced here, this little overhang that we've got here, this is called a 3' extension. Ooh, and I've realised that my picture doesn't demonstrate it brilliantly, but the 3' strand should be shorter than this 3' strand here. Let me extend though. Oh no, that'll ruin that picture because I've drawn them perfect. Oh, I suppose I can extend them a little bit. Okay, so remember, until we reached the chi sequence, we were degrading both strands, okay? So we were cutting them both back, we were trimming both of them, okay? So they are now shorter than these original ones that we had here, okay? But now, once we reach the chi sequence within this 3' ended red strand, we then stopped degrading the red strand, but continued cutting back the 5' ended strand, which is the turquoise one in this case, and therefore we've produced this 3' extension, this overhang of the 3' strand, where it's got no complementary turquoise strand attached to it. Okay, right. Now what's going to happen is this overhang is going to invade our intact chromosome here. Okay, so we're going to get a process known as strand invasion now. Okay, and this is going to start the process of homologous recombination. Okay, so effectively, remember what we've got here. This red strand is complementary to this orange strand. This red strand has the same sequence as this blue strand here. Okay, we've trimmed it back a bit, so now this extension here, this 3' extension, will be complementary to some portion of the orange strand here. So what we're now going to do is we're going to detach that portion of the orange strand away from the blue strand, and we're then going to attach it onto the red strand. Okay, so let me show you this. So we'll start by redrawing the red strand and the turquoise strand. So here is our red strand, and here is our turquoise strand. So that hasn't changed. Okay, we've got our 3' extension here. Now what's going to happen is we're going to have our orange strand here, and now we're going to pull it out, and we're going to attach it onto that red strand like so. Okay, now remember we have trimmed back our 3' ended strand a little bit, okay, before we reach the chi sequence. So the red strand is now shorter than the orange strand, so the portion that is the 3' extension will be complementary to some portion of the orange strand back here, which means that there's more orange strand here that isn't attached to the red strand. Okay, then of course we continue the orange strand on with its turquoise bit here. Then the blue strand will have been sort of debunked, basically. It's been had its orange strand torn away for part of it. Okay, so we've opened up the connection between the orange and the blue, and we've said, okay, I don't want the orange strand now here connected to the blue strand. We'll connect it instead to this 3' extension on the red strand. Okay, and that is called strand invasion because we are invading this double-stranded piece of DNA. Okay, right. So now what you're going to do is you're going to undergo a process called branch migration, and I'll put this here. Okay, so basically you are going to extend this connection that we have between that red strand and that orange strand. Okay, so this is the principle of what we're going to do. We're going to imagine pulling this turquoise little fragment we've got here away from the red strand. So imagine peeling this turquoise strand back off there to expose more of this red strand here. Okay, then what we're going to do is peel the orange strand off the blue strand and attach it instead to this red strand. So let's start by showing that. Okay, so this is why it's called branch migration, because we are effectively migrating this point where the strands are intertwining or branching. Okay, right, so now our orange strand is going to be making a much larger contact with our red strand here. Okay, like so. So we've peeled this turquoise strand off that red strand, and we've peeled the orange strand off the blue strand, and we've reattached the orange strand onto the red strand there. Okay, now that's going to leave firstly this turquoise strand here with nothing to bind to. It's also going to leave this blue strand a little friendless. Okay, so here's the blue strand here. Like so, still attached to the end portion of this orange and turquoise strand up here. Okay, but now what we're going to do is we're going to bring this turquoise strand here, which is dangling around now. Okay, its end is still attached to the red bit, but we've peeled this bit back, so this bit's now going to be flapping around, basically. So what we're going to now do is attach that onto this blue portion down here. Okay, like so. So, effectively we are saying, peel this turquoise strand off this red strand and attach it instead to the blue strand, meanwhile peeling the orange strand off the blue strand and then attach that to the red strand. So effectively swapping the strands, okay? And this structure that we have created here, this is what is known as a holiday junction, okay, or a holiday intermediate. Okay, so we've got what's called a holiday intermediate, named after the geneticist or biochemist, whoever came up with this first. Okay, whoever imagined this first, I think it's named after. It's named after a scientist anyway. Okay, right. There's another way of drawing these holiday interactions or holiday intermediates, which is going to be helpful to us. Okay, so what you can do is you can draw the two strands that are coming in here. So we'll start by drawing this orange and blue strand. Okay, so we'll start by drawing, this picture is going to be useful to us. I think this picture is more simple to understand, but this other picture is going to be useful to us because this is actually what it looks like in the cell. So I'm going to draw this other picture. So here is the orange strand and the blue strand, okay, which were originally connected to each other. We're now separating them off, okay, so we're opening up this gap in between them, okay, like so. Now, eventually they will rejoin together, so the orange strand will turn into the turquoise strand here, and this will reconverge with the blue strand. And remember, we're just at the start of our chromosomes, so these will go on and on and on and on. Okay, let me bring this up. So I hope you can appreciate that all I've done is taken this orange strand, which has the turquoise portion, and the blue strand out, and they've opened up at this middle portion. Okay, we're then bringing in this other strand, this red turquoise strand here, which remember was this tiny little fragment that we produced because of the broken replication fork. Okay, and we're bringing this in here, okay, and we're now attaching it onto the orange and the blue strand in this opened portion here. So let's show this. So here is the red strand here, which is getting attached onto the orange strand here, and here then is the turquoise strand, which is getting attached onto the blue strand here. And remember, the turquoise strand will be slightly shorter than the red strand because we produced that 3' extension. In addition, the red strand will be shorter than the orange strand because, remember, we cut the red strand back when we first attached this complex onto the end here. We cut both the red and the turquoise strand back. Okay, so they're now shorter than the orange strand, even though they originally started off the same length as it. Okay, right. So this is actually what these holiday intermediates are going to look like in the cell. Okay, and we'll come back to this picture later. Okay, now what you're going to do is you're going to finish this process off. So we've got the holiday intermediate at the moment. Okay, that's useless. We want to actually create a replication fork. So what are we going to now do? Well, we're going to get an enzyme in here to cut the strands in the holiday intermediate. So where are we going to cut? Well, I think it's easier to understand on this picture than this picture. So we'll start with this picture, then we'll go to this picture. Okay, so we're going to cut this turquoise strand, and we're going to cut this orange strand, and then the idea is we're going to bring this little fragment of the turquoise strand here that we've detached off from the rest of the turquoise strand over here, and we're going to bring it onto this blue strand and attach it up to this little fragment of the orange strand here. So let's draw that out. Okay, so let's start with that little fragment of the orange strand there. Okay, so this portion here is this portion here. Then we've got this little portion of the turquoise strand that is now attached onto the blue strand. So this portion here is now going to be attached with this orange strand. So we're going to firstly cut these strands, and then we're going to ligate this turquoise strand here with this orange strand. So this is a process of both cleavage and then ligation. Okay, so we're going to get cleavage and then ligation of the two strands. Okay, and then we've got this little orange and then turquoise strand here. We've then got our blue strand here. Okay, like so. Then we're going to do the same thing over here. We're going to take this turquoise strand here, and we're going to attach it to this orange portion here. Okay, so now we've got this turquoise strand here, and we've got this orange portion here, like so, which then goes on to become the rest of this turquoise strand here. Okay, which will be still attached to this blue portion here. Okay, with the rest of the chromosome going off there for potentially, yonks. Okay, then the one last strand we've missed is the red strand. The red strand will be bound to this turquoise strand and this orange strand here. And now, there you have it. We have recreated a replication fork. Okay, we can now bring in our replication machinery to continue this on. So we have reassembled a replication fork. Now, it is more complicated than I originally said there. Okay, this was a basic picture of what the basic concept was, that we were going to reopen this new strand and then put these two strands here. Okay, it's more complicated than that. We're going to swap bits around basically like so. Okay, that's why it's called homologous recombination, because we're moving bits of DNA all over the place. But notice the bits of DNA that are being moved are identical. They're genetically identical. So it truly is homologous recombination. Okay, right. So that's the picture in terms of this nice diagrammatic picture, but what does it look like in terms of this picture, which is actually what it looks more like when we look at what is happening within the cell? Okay, well, where are we going to put these cuts? So we cut this orange and turquoise strand here. So that strand here, that is this strand here. So we must be cutting somewhere around here. Okay, so we'll put our ligation there. Okay, where else are we going to cut? Sorry, that's not a ligation, that's a cleavage site. Where else are we going to cut? We're going to cut this little turquoise strand here. So that must be somewhere around there, and indeed that's what we're going to do. We're going to cut here, and then what we're going to do is we're going to attach this orange strand here to this turquoise strand here. So this portion and this portion are going to get attached to produce one strand here. Okay, and then this orange strand here is going to get attached to this turquoise strand here to produce the other strand there, and that's going to produce us this same replication fork here. So let's just draw this out. Okay, so keeping the picture now analogous to this second picture, here is our blue strand once again, which will fold around like this, and then be later attached onto our turquoise strand, which is down here. Okay, here is this orange strand that goes up here. Okay, now where have we cut? I'm going to have to be careful now. Okay, so we've cut there, so we now have this orange strand ligated up to this turquoise strand here, coming down like so. And then we've got the red strand bound there. Okay, then we've also got this initial portion of the orange portion here, now ligated up to this turquoise portion here. Okay, like so. So that is how this picture translates into something that looks more like what happens in the cell. Okay, and hopefully you can see that it's the exact same picture. For instance, here's this blue strand, here's this blue strand. It has an orange and then a turquoise portion, an orange and then a turquoise portion bound to it. The other strand, we've got turquoise, orange, turquoise, turquoise, orange, turquoise, with the red strand bound there. Okay, so that now is our big picture of homologous recombination for replication fork reactivation. And by the way, replication fork reactivation is often abbreviated to RFR. Okay, what we're going to do in the next video is we're going to discuss the proteins that are involved in doing this in Escherichia coli.